Hi, welcome to C2.2 Neural Signalling for the IB Biology new spec, the first exams of which are in 2025. This is just a reminder of the IB Biology guide for SL, so you can refer back to this as and when you need to, and the HL guide if you need it for reference at any point. This is the key terminology for this entire chapter. You'll find the SL key terminology in green and the HL key terminology in blue on this slide. Throughout the video, all of the key terms will be highlighted in red, so you'll be able to see which terms are key terminology if you want to make flashcards or use those to guide your revision. So here we have the two branches of the nervous system. We've got the central nervous system made up of the brain and the spinal cord, and we've got the peripheral nervous system made up of all of the rest of the neurons, so usually the sensory and motor neurons. You can see in the diagram that the central nervous system is in red and the peripheral nervous system is highlighted in blue. This is the structure of a neuron, so you must be able to draw this and label it for the exam. So you can see you've got the cell body here, you've got the long axon through the middle, and you've got the axon terminals here that will synapse with the next neuron. You've also got these cells here. These are Schwann cells. You can see there's 5 of them here. Now they make up the myelin sheath, and the myelin sheath helps to insulate the axon, and it also helps to speed up the transmission of nerve impulses, which is really important because the quicker the impulse, the quicker we can respond. There's also sensory and motor neurons. So the sensory neurons are the neurons that detect any sort of external stimuli via receptors, usually in the sense organs, and they take the nerve impulses to the central nervous system. The central nervous system then coordinates the response and sends the response via the motor neurons to something called effectors. Now, effectors are usually either muscles or glands, and they cause the reaction to the initial stimulus. Okay, so at rest, the axon, so the long part of your neuron, will be negatively charged on the inside and positively charged on the outside, and you need to be able to explain why that's the case. And the reason that's the case is because if we think about the membrane of the axon just along here and also along here, you'll find 3 different proteins embedded. You have a sodium-potassium pump in light blue, you have the sodium pump here, the sodium channel, in purple, and the potassium channel in yellow. Now, at rest, so when there are no nerve impulses traveling along the axon, this sodium-potassium pump will constantly be pumping out 3 sodium ions out of the axon for every 2 potassium ions that are pumped in. Now, that means that we're pumping out more positively charged sodium ions than we are the positively charged potassium ions going in. So therefore, we end up with this positive charge on the outside of the membrane and a negative charge on the inside because you've got more ions on the outside. You've pumped more out than you have in. Sodium and potassium channels at this point are closed. Now you will sometimes read that they are kind of ever so slightly open and there may be some leakage of ions, which is true, but they are mostly closed. There's very little movement of sodium and potassium ions through those channels, and the membrane potential at this stage is minus 70 millivolts. Now this information is in a green box. This information here is in a blue box because this is HL information. So we need to know how that nerve impulse is passed right down this whole axon, and in order to do that we need to depolarize the membrane. We're going to depolarize the membrane in sections, so one section and then the next section and then the next section. And when depolarization happens, the sodium channels are going to open. So they were previously closed at rest. They're now going to open in depolarization, and when they open, the sodium ions are going to flood in to the axon. So we were previously pumping them out from their low concentration to their high concentration, and now when we open that sodium channel, they're going to flood in. When they flood in, they cause the inside of the membrane to become positively charged. So you can see this depolarized region here where the charge across the membrane has switched. So this is polarized, this is at rest. This area hasn't been depolarized yet. This area has been depolarized, so the charge has switched, and we can see that the depolarization is going to move to the right here. So therefore, this area of depolarization will move to the next section of the axon. We've also got the threshold potential. Now, the threshold potential is the potential that must be reached in order for the action potential to be sent. So, for example, if you held out both of your hands and closed your eyes, and in one hand I placed a feather and in the other hand I placed a brick, the brick would exert enough pressure for an action potential to be sent because it reaches the threshold. There is enough stimulus to reach the threshold. The feather, you might not feel it because it doesn't have enough weight to exert enough pressure on your skin for a nerve impulse to be sent. So the threshold has to be reached for it to be sent. And the all or nothing principle suggests that if the threshold is released, then an action potential is always sent, and if it's not reached, it's not sent. There's no half action potential or part action potential. It's either sent or not. So it's all or nothing. Now, once we've depolarized the membrane, we need to repolarize it back to resting. So you can see that you've got your area of depolarization here. Your wave of depolarization is moving to the right into this area that is at rest. So what then happens if we look at this diagram? We can see that in the area that is repolarizing, the charge has switched again, and the charge has switched thanks to the fact that the potassium channels open and potassium ions flood out of the axon to try and restore resting potential. So these are the points that will get you marks in the exam. Lots of textbooks and lots of sources go into a bit more detail, but this is what will get you the marks. Once we've repolarized this area, it will return to resting potential after it has hyperpolarized. So there's this short space of time where we say that this membrane here, this part of the membrane, would be hyperpolarized. And what happens there is that more potassium ions flood out than are needed to restore resting potential. And this means that this area cannot be depolarized. So you've got your area of depolarization. That wave is going to move to the right. We don't want it to be able to move to the left because that means that this depolarization could just ping backwards and forwards within this axon and never get to where it needs to be. So we only want it to be able to move in this one direction. So this area is hyperpolarized. So it repolarizes, and during that time it hyperpolarizes, and it means that this cannot be depolarized, so the wave only moves in one direction. This is also called the refractory period as well. Actually, you can see there it's called the refractory period. Hyperpolarization and the refractory period are the same thing. Okay, so sticking with the HL content, there's also this action potential spike, which looks quite confusing, but once you've gone through the stages of the action potential, this actually makes quite a lot of sense. So you can see here at rest, we're at minus 70 volts. So this is the axon at rest. We can see here the depolarization is occurring, and so therefore the membrane potential is going to increase until about plus 40. At that point the membrane is depolarized, and then it repolarizes, and you can see this really nicely highlighted on this action potential spike that the refractory period is where the membrane potential becomes more negative than at rest. So in this point here, the membrane cannot be depolarized, and then it returns to rest, and once it's returned to the resting state, it can be depolarized. You've also got the threshold potential here, which is about minus 55 millivolts. Now there's this little section that says failed initiations. This means that there was a stimulus, but the stimulus didn't reach the threshold. So we haven't reached that threshold, so our action potential was not fired. At this point, it has reached the threshold, so this one is fired. These are just the failed ones that didn't work because it didn't reach the threshold. Finally, for this section, we've got saltatory conduction. So saltatory conduction is where local currents are set up between the nodes of Ranvier. So you can see here that you have your sodium ions in the area of depolarization, and they diffuse into this area that is at rest, and this helps to set up local currents. Now, we talked earlier about how these Schwann cells and the myelin sheath will allow for the nerve impulses to move more quickly along the axon. And the reason that this happens is because only the naked parts of the axon can be depolarized. So where the Schwann cell is, you cannot actually depolarize that axon. So it means that your waves of depolarization jump from this bit of axon and it jumps to the node of Ranvier in between the Schwann cell, and then it jumps again and it jumps again, and it means that increases the speed of transmission because you don't need this wave of depolarization to move all the way down the axon. It can just jump from node to node until it gets to the axon terminal, and we call this saltatory conduction. Okay, back to some SL content. So here we have a really nice diagram of a synapse. So this is your axon terminal. So this is the end bit that on the previous diagram was right to the right hand side of your neuron. Now this is going to be our presynaptic neuron. We have our synaptic cleft here, this little gap in between the neurons, and then we have the postsynaptic neuron here. So this is our second neuron that we're trying to pass the signal to. We also have some vesicles in here. We have calcium channels that we haven't seen before. We've got something called neurotransmitter, and we've got some ion channel receptors here. So in synaptic transmission, the nerve impulse is going to arrive at the end of the axon. And when it arrives at the end of the axon, it causes these calcium channels to open. Once the calcium channels open, calcium is going to flood in to this synaptic bulb. So we call this section the synaptic bulb. So the calcium floods in. And this triggers all of these vesicles here, the blue vesicles, to move towards this presynaptic membrane. Now the vesicles contain something called neurotransmitter, and this is the chemical that is going to take our signal from the presynaptic membrane to the postsynaptic membrane. So the calcium is flooded in. That triggers these vesicles to move towards the presynaptic membrane, and you can see that they're fused with the membrane, and when they fuse with the membrane, this causes them to empty their neurotransmitter into this synaptic cleft. The neurotransmitter is then going to diffuse across the synaptic cleft into this postsynaptic neuron, and it attaches to some receptors on the postsynaptic neuron. And when it does that, it causes these sodium channels to open. So sodium floods in, and we learnt previously that when sodium floods in, this causes depolarization and triggers the action potential in the second neuron. So this is a really nice extended response question. If you can learn this process, you could get 5, 6, maybe even 7 marks for that. Now, a really common neurotransmitter that we need to know about is one called acetylcholine, and it's the neurotransmitter that we find at neuromuscular junctions. So it's released from neurons that synapse with a muscle and then cause that muscle to contract. So the acetylcholine is within this synaptic cleft and it binds to its receptors. Now, once it's bound to the receptors, and that because the action potential arrived, we need it to leave again because we don't want it to continue stay bound to the receptor, because that would mean that in the muscle there would be continuous contraction, and we don't want that to happen. So therefore, the acetylcholine is broken down by an enzyme called acetylcholinesterase, and the products of this breakdown can then be absorbed back into the synaptic bulb and repackaged into these vesicles to be used again. Just for a bit of HL content here. So acetylcholine is an excitatory neurotransmitter, and that causes more action potentials to be fired. And there are some other neurotransmitters, GABA is an example, that are inhibitory neurotransmitters. So these prevent the action potentials from being fired. And there's a number of ways they can do this. So for example, some might block the receptor from being able to bind to another neurotransmitter, and therefore the sodium channels won't be able to open. Okay, so we have some more HL content here. So we've got exogenous chemicals and endogenous chemicals. Now, exogenous chemicals are chemicals that are produced outside of the body, and endogenous chemicals are chemicals that are produced inside the body. And both of these can have an effect at synapses. So examples of exogenous chemicals might be some drugs, some medications. Endogenous chemicals are obviously natural compounds and molecules that are produced in the body. So our examples are neonicotinoid insecticides, and these are chemically similar to nicotine. However, they attach to the receptor, the same receptor that the acetylcholine neurotransmitter attaches to, and this blocks the receptor. So if our acetylcholine cannot bind to the receptor, it means that the action potential cannot be fired in that second neuron, and therefore the muscle won't contract, and that leads to muscle paralysis and death of an insect. So this is specifically to try and kill insects, not people. Cocaine, obviously an illegal drug. This prevents dopamine from being removed from the synapse. So we talked about neurotransmitters being broken down after they've done their job in the synapse. Now, dopamine is a neurotransmitter that causes feelings of pleasure. So cocaine stops that dopamine from being removed from the synapse. It means it can't be broken down and taken away. So the dopamine remains in the synapse and continues to bind to the receptor, and the action potentials that are going to cause feelings of pleasure continue to be fired if you take cocaine. And cocaine is an addictive drug as well. Summation is the effect of the excitatory and inhibitory neurotransmitters together. So it's the sum of the signals, and it's whether that the sum of those signals results in an action potential being fired. So sometimes the excitatory neurotransmitters, there'll be more of that and your impulse will be fired. Sometimes there'll be more inhibitory neurotransmitter and therefore the impulse won't be fired. There's lots of scope for some nice exam questions there, because there's lots of different examples of drugs and chemicals that may increase or decrease the amount of different neurotransmitters. We've got just a little graph there that talks about APSP and IPSP, the excitatory postsynaptic potential and the inhibitory postsynaptic potential, which might be quite useful to have a look at. Okay, finally, we've got the perception of pain. So we need to know that sensory receptors detect external stimuli. You find sensory receptors in sense organs generally. So you can see here that we've got thermoreceptors that can detect temperature. That's useful because obviously we don't want things to get too hot and burn us. We've got nociceptors that detect tissue damage and pain. Electromagnetic receptors that detect electric current and magnetic fields. Photoreceptors in the eye, obviously. Baroreceptors, which detect blood pressure, and chemoreceptors, which detect chemicals. So a really nice example is detecting taste on the tongue or smell in the nose. And sensory receptors can either be free nerve endings or specialized neurons that you find within the sense organs. And those are your examples there. And then finally, we have consciousness as an emergent property. Now, emergent properties crops up throughout the new IB specification, and an emergent property is a characteristic that an entity gains when it becomes part of a bigger system. So something that happens when you put all of the pieces of the jigsaw together, and then the emergent property is the result of that jigsaw coming together. So reductionism is understanding the more simple parts of an organism being crucial to understanding the working of the organism itself, whereas emergence is the fact that the whole is greater than the sum of its parts. So a nice example would be, obviously not a living example, but all of the parts of a car are useless until you put them together to make a car which can then transport you from A to B. And then finally, the interaction of all the systems within organisms can lead to consciousness. So within humans, when everything within the body interacts, it leads to consciousness. And consciousness is an emergent property because that can't happen if all of those systems aren't working together successfully. This brings us to the end of the video. I hope it was useful. The revision sheets are available if you want to take all of this information, change it into your own words and have a revision sheet of your words, but with the correct information that will get you marks in the exam.